This series of videos is intended for researchers who already have knowledge of PSC culture techniques and are interested in trying or improving their PSC aggregate suspension culture in the PBS Mini Bioreactors. This is the first in a multi-part series, and the topics in this video are choosing the right PSC culture medium and reagents, and the importance of having a high-quality starting cell population to inoculate into your 3D culture. The quality of the cells inoculated into the bioreactor determines how successful your 3D culture will be. Before 3D culture, it's necessary to expand cells in 2D. We refer to this as the 2D seed train, and it serves four purposes. First, to produce a sufficient quantity of cells to inoculate into larger scale cultures like a bioreactor. Next, to conserve master cell bank resources by requiring fewer cells to be thawed. Also, to allow cells to recover from cryopreservation in a less stressful environment, leading to a reduced lag time in 3D culture. And finally, to avoid multiple passages in 3D culture, which has been reported to have the potential to introduce genomic instability.
Here are our recommended 2D seed train culture parameters. They have been validated in all types of 2D culture vessels, from well plates to multi-layer flasks. This protocol has been tested with multiple pluripotent stem cell lines, cell culture media, extracellular matrix coating substrates, and dissociation enzymes, and consistently produces high-quality PSCs ready for inoculation into 3D culture. Our baseline protocol that we use as a starting point for optimization uses mTeaser1, Matrigel, and Accutase. We always include 10 micromolar ROCK inhibitor in all of our passaging reagents. ROCK inhibitor helps cell survival during passaging, helps to form aggregates, and preserves pluripotency. We use ROCK inhibitor in all harvest and passage reagents except for DPBS. ROCK inhibitor is removed from culture during the first medium exchange. It's not necessary to use these exact materials for your process. The best materials to use are the ones that work best with your cell line and meet your constraints, such as cost, supply chain availability, quality certification, and whether or not they're chemically defined. We've screened many combinations of PSC culture reagents, and none have been incompatible with our 2D seed train into 3D aggregate culture protocol. If you're not having success with your chosen reagents, try testing others. When beginning process development studies, it's best to screen a wide range of options and see what works best with your cells. As illustrated in this graph, every commercially available medium we've tried has been able to support PSC aggregation and expansion in 3D culture. When first starting out, we recommend trying a few different options to see what works best with your cell line according to your downstream goals and constraints. Based on your application, your 2D seed train protocol may not follow our exact recommendations, but before moving on to 3D culture, it must be well characterized, as in you know the expected growth rate, morphology, and pH range of your culture, reproducible, as in you see the same results when the process is performed in exactly the same way, and robust, as in you see similar results when some non-key variables are changed, such as operator, lot number of material, or cryovial from within the same cell bank. It's imperative that the 2D seed train is well understood and well controlled before advancing to 3D culture, so that results from experiments in 3D culture can be fairly evaluated and compared to one another. These are the expected values for cell density and cell fold expansion if the recommended 2D seed train protocol is followed. Different cell lines will perform differently, but these numbers represent the average plus or minus standard deviation of 24 independent runs using two different cell lines. The population doubling time in the first passage is longer while cells are recovering from cryopreservation. Using our recommended protocol, the cumulative population doublings over both passages in the 2D seed train are remarkably consistent, demonstrating the protocol's reproducibility. The morphology of PSCs in 2D culture should be tightly packed, distinct colonies with defined edges. At the time of harvest and passage, cells should be in their exponential growth phase, not overconfluent, and in medium at a physiological pH range, around 7.0 to 7.4. By overconfluent, we mean greater than 90% of the surface area of the flask is covered, and cells are starting to grow on top of each other. Characterization studies should be performed to determine what the pH and cell number ranges are for a given cell type and process. Cells may be inoculated into the PBS Mini directly after thawing, but not all cell lines and cell banks will work with this method. It's best to start with a 2D seed train and get familiar with 3D culture techniques before attempting the more tricky direct thaw method. If you do attempt the direct thaw method, your cell bank should be well characterized in 2D culture and determined to be of high quality. Pluripotent stem cells are adherent cells, and when they're inoculated into the PBS Mini, they spontaneously form aggregates. The homogeneity of the aggregates is of critical importance to efficient expansion and downstream differentiation, and the medium exchange method has a massive effect on aggregate homogeneity. Medium exchange via continuous perfusion allows for the most uniform aggregates while maintaining stable nutrient and metabolite levels. And perfusion is possible in the PBS 0.5 vessel using an integrated sterile perfusion cap. More details on perfusion later in the series. In summary, a 2D seed train is recommended in order to produce a sufficient quantity of high-quality PSCs to inoculate into the bioreactor. By following our recommended protocol and optimizing it based on your cells and constraints, you can ensure your 2D seed train is well characterized, reproducible, and robust. In the next video, we'll be covering recommendations for inoculating cells into 3D culture, including harvesting from 2D, preparing an inoculum, preparing the mini vessel, and 3D inoculation strategy. Thank you for watching, and if you have any questions, please contact us through the link in the description.